Hi everyone, I'm Arsalan Riaz, an undergraduate from SCS University and a research fellow at Precision Medicine Lab, Pakistan. I'm also the Vice President of RSG Pakistan, which is the Pakistan chapter of ISCB SC. Thanks to the SCS team for providing me the opportunity to represent my research work. The talk is titled Computational Classification of Pathological Stages of Head and Neck Squamous Cell Carcinoma Using DNA Methylation. I have no conflict of interest for this research. This project revolves around the head and neck cancer, which is the sixth leading cause of cancer, especially in South Asian country, including my country Pakistan. Research has shown that DNA methylation is an important driver of many of the distinct stages of cancer. In previous studies, two-stage classifiers have been developed to predict the early and late stages of liver carcinoma using differentially methylated CpG sites. But however, there is still a gap for building four-stage classification model for predicting pathological stage from stage 1 to stage 4A using not just differentially methylated CpG sites but to include all those CpG sites that are found to be associated with the mutated genes in HNSCC, which are often neglected due to the computational expense and time. The aim of the study was to identify key CpG sites and build machine learning classifiers to predict pathological stages of HNSCC. To achieve this, we aggressively applied Baruta algorithm to about 485,000 CpG sites of 108 patients ranging from stage 1 to stage 4A of HNSCC with 27 samples per stage to avoid the class bias. Then, machine learning models were built using random forest, support vector machine, k-nearest neighbors classifiers. After this, we performed network analysis and enrichment analysis on the genes associated with the identified CpG sites using Baruta algorithm to understand the biological significance of the genes. Before proceeding to the results, we also need to understand the pre-process step. After compiling the methylation data of 108 patients, only those CpG sites were taken into studies which were associated with genes mutated in HNSCC. So, after missing values imputations, 294,000 CpG sites were left, which were then divided into 148 sets with 2000 CpG sites per set, except for the last one, which contained 603 CpG sites. For each set, Baruta algorithm was applied separately and the resultant CpG sites were combined from each set and again were passed through the same algorithm, that is the Baruta algorithm. And at the end, we extracted 68 CpG sites through the algorithm by calculating the importance level based on comparative z-score of the shadow and the original attributes. When we used these 68 CpG sites as features in machine learning classifiers, we achieved an accuracy of 90.68% with random forest classifier, which is very significant as compared to the support vector machine with both kernels and k-nearest neighbor classifier, which achieved an accuracy of 71 with support vector machine radial kernel and 68% with support vector machine linear kernel and 56% with k-nearest classifier, which is very low as compared to the random forest. That can also be seen in the box plot and the confusion matrices. On feature contribution analysis in the best classifier, that is the random forest, we observe the contribution of most CpG sites for stage 4A and least for the stage 1. Consistent with the fact that tumor progression is attributed to the accumulation of alteration over time deriving the course of the disease. These CpG sites that are prioritized using the Baruta algorithm are associated with 67 genes. Network analysis showed that LMNA, HIST3H3, and CRK are the hub genes in the human PPI network of 63 proteins encoded by the genes as shown in the network of 2566 nodes and 3374 edges. Interaction for the remaining 4 genes out of 67 were not found. We also found 18 densely connected proteins using M-code. Enrichment analysis also showed that the genes were enriched in Go terms associated with structural development, cell differentiation and proliferation, signal transduction, metabolic and biosynthetic processes, while some genes were also involved in cancer-related pathways like MAPK and WNT pathways, signaling pathways, and also apoptosis. In conclusion, we identified 68 CpG sites as potential features using the Baruta algorithm, and a random forest classifiers can be used to predict the pathological stages of HNSCC with significant accuracy, which demonstrates the capabilities of the Baruta algorithm on larger datasets like DNA methylation datasets. In future, an integrated omics analysis of these findings will help us narrow down the list of further genes or candidates, and then the role of those candidates will be needed to be experimentally validated using in vitro or in vivo models. In the end, I would like to thank the Precision Medicine Lab, our funding bodies, that is the Higher Education Commission of Pakistan, National Center of Big Data and Cloud Computing, SCS University, and Rahman Medical Institute for funding this project. I would also like to thank the ISCB SC for putting up an amazing conference for students at the platform of SCS 2021. You can also contact me at muhammad.arsalan@ccos.edu.pk. Thank you.
Thank you so much. Another amazing work on HNSCC. So we have one question from, if I can pronounce it rightly, Romana from University of Oslo, and she asks that what exactly does this Baruta algorithm do? So I suppose we lost connection. Uh, yeah, he's back.
Can everyone hear me?
Yeah, now we can.
What it does, it, it replicates the original attributes, and then these copied attributes are shuffled across. Then the maximum z-score using random forest, and on the basis of these z-scores, it classifies to which features are important as compared to others. And on the basis of this maximum z-score, it eliminates less important features. And in the end, we get a subset of important attributes which can be used in further machine learning models. I hope it can answer the questions. It was the simplistic explanation for this point.
Okay. Thank you. I hope, Romana, it answers your question. If not, you can again ask him. So the next question is by Pradeep Irenti. He is also a member of ISCB, and he asked that how did you generate the PPI network or the source of the network? What method is used for the network analysis part? So.
Okay. So the main source of the PPI network is BioGrid, and we know that it is whole consortium of networks which is available on BioGrids. So what we did, we used our genes and looked where our genes means the genes that were associated with the identified CPG sites. So we took those genes and looked for interactions in the huge networks of human PPI, which was taken from the biogrid. So we just used our programming to look for interactions that involve our genes, which are mostly 67 genes, and then we used cytoscape to visualize this network.
Okay. So I think Pradeep, it answers your question. I also have one question for you, Riaz. That could you tell something about? So you have used Boruta algorithm. So is there any shortcomings of this algorithm or drawbacks of this algorithm for users?
Yes, of course. Since we are dealing with computations, and it will be obvious that there will be some sort of shortcomings. The main shortcoming that I feel that the sample size. We have taken 108 samples, and which in each stage have 27 per stage. So it will be nicer to have more sample size. And one more drawback can be that since the data was taken from TCGA, and if you look at the demographics of for TCGA, these are mostly related, the samples are mostly related to white. We really wanted that if we had a sample size from most of the demographics like Asians, Europe, and Americans and Black as well. But we will stick to to the white sample because available at that point.